Does DNA really help a taxonomy? Is morphology insufficient to define species? According to syndromes, the study of systematic taxonomy based on morphological characteristics feature is very essential in classification system. In recent times, the experts on this basic taxonomy is significantly declining and that made to seek some alternatives. Moreover, basic taxonomy may possibly be difficult, time consuming and restricted with special group of organisms for a specialist who practiced this classical study. This also cost more manpower with flawless expertise in the taxonomic study. DNA based analyses are primarily supplementary or complementary and needs data on classical taxonomy in literatures to compare and confirm. Without existing taxonomical data, DNA taxonomy alone might not resolve the problem. There should be a combined efforts to find solutions in this area of research. According to Mohad Shabir, the morphology is useful in identifying species, whereas DNA help in confirmation of the species identity as well as studying phylogenetic relationship among species. According to A.K. Jaswar, morphological taxonomy is the first requirement. Without classical taxonomy, it is impossible to carry out molecular taxonomy. It is like a tire, you do not know which car it belongs to. That correct identification is done by morphological characters and then the specimen is barcoded and the barcode is submitted. If someone identifying based on barcode which is present in gene bank and the one who has submitted has missed correct identifications, the exercise goes wrong. Therefore, it is requested to go for correct identifications seriously, then only barcode and submit. If I have identified a specimen correctly and I try to match barcode for my specimens with already present in gene bank is wrong, then I will land with many confusions like I will think mine is a new species, etc. It has to be done carefully, very carefully. According to the Williams, an examples where DNA can help is when dealing with animal parts, for example, dried shark fins. DNA barcoding has been shown to be able to accurately distinguish 99% of the samples of the shark and race to the species. When dealing with dried sharks, fins where morphology of the fin is difficult, DNA barcoding provides a method for determining species compositions which was previously lacking. So yes, in some cases DNA is very helpful for taxonomy. It is a tool and like all tools it can be misused and a lot of peoples do have misused these tools. But in the right stand, but in the right hands, it is a powerful tool which should not be ignored but instead used in concert with classical taxonomic method. According to the Ram Nivas, only DNA data cannot help to know about the species level of any organisms. The sequencing data with the specific primers might be helpful for this. According to the Nazia, first we have to check all the morphological characteristics including media, the temperature of the growth, light intensity in the case of fungus, structural morphology through microscopy and then biochemical characteristics, then molecular characterizations.